Hello guys, today we'll be solving Cambridge IGCSE Biology Paper 6 Alternative to Practical October-November 20 Paper 61. Question number 1. The enzyme lipase catalyzes the breakdown of fats into fatty acids and glycerol. A student investigated how the concentration of the lipase affected the breakdown of fat in milk. An increase in the concentration of fatty acid in the milk will change the pH of the milk. The student used a pH indicator, bromothymol blue, and observed the color change of the indicator in milk and lipase solution. Table 1.1 shows the color of bromothymol blue indicator at different pH values. So we can see the pH value 6 provides us yellow, 7 provides us green, and 8 provides us blue color. Step 1. 4 test tubes were labeled L1 to L4. Step 2. Solutions containing different concentration of lipase enzyme were made using the volumes of 2% lipase solution and distilled water shown in table 1.2. L1. Volume of 2% lipase solution 3 cm cube. Volume of distilled water added 0. So percentage concentration of the lipase solution is 2.0. So similarly, L3 is using 0.75 volume of 2% lipase solution. Volume of distilled water is being used 2.25 cm cube to make it a final volume of 3 cm cube, and the concentration is unknown. Okay, calculate the percentage concentration of lipase solution in test tube L3 using the information in table 1.2. Alright, so basically we can use the formula here. V1 C1 volume into concentration is equals to V2 into C2. So V2 C2 is the second concentration. Now initially we already know the volume that we are using for the 2% lipase solution, which is 0.75, and the concentration is 2%. The final volume that we want to achieve is 3 cm cube, but we don't know what is the final concentration is, so C2 is unknown. So let's make C2 our subject. 0.75 into 2 divided by 3 equals to 0.5 is the answer. So obviously 0.5% will be our final answer. Step 3. Another 4 test tubes were labeled M1 to M4. Step 4. 3 different substances were added to each test tube M1 to M4. 5 drops of bromothymol blue indicator, 2 cm cube of sodium carbonate solution, 2 cm cube of milk. The student observed that the contents of the test tube M1 to M4 were all blue. Because we added the sodium carbonate solution, it is alkaline. So initially every one of them is turning blue because of the presence of sodium carbonate. Step 5. All of the test tubes were put into a water bath containing warm water and left for 5 minutes. Step 6. After 5 minutes, test tube M1 and L1 were removed from the water bath. Step 7. The contents of the test tube M1 was poured into L1 and a stop clock started. So basically we put into the warm water bath so that we can calibrate them to the same temperature and then basically start the experiment. Step 8. The color of the bromothymol blue in test tube L1 was observed. The time taken for the bromothymol blue indicator to become yellow was recorded. If the color had not changed to yellow in 5 minutes, the result was recorded as greater than 300. Step 9. Step 6, 7, 8 were repeated for test tube M2, L2, M3, L3, and M4, L4. The time for the step 8 and step 9 are shown in figure 1.1. Prepare the table for the results. Convert the time shown in figure 1.1 to seconds and record them in our table. So basically in this kind of table we will have to record the test tubes that we are using. So initially to recognize the particular test tube that we are using, we can definitely use the L1, L2, L3 and L4 for our recognition because they are the ones that are mentioned in this figure 1.1. So first of all, test tube. and the other one will be time slash second needed for it to change color. So first of all, answer for L1, it took 25 seconds, so we'll just simply write down 25. For L2, it took 38 seconds, so we're just directly going to write down 38. For L3, it took 1 minute 17 seconds, so 1 minute is equals to 60 seconds plus 17 second is equals to 77 seconds. L4, we can see no change at 5 minutes, so we are definitely going to write down here greater than 300. State a conclusion for these results. Now we can say as the concentration of the enzyme increases, the time to change color decreases. B1. Identify the control in this investigation and explain why a control was used. So basically our control in this investigation was L4 to show that lipase is causing the change of color in the indicator. As you can see in L4, we did not use any lipase. So basically we proved that lipase was the main reason why we are having this particular chemical reaction. Using the information in table 1.1 and the results, estimate the pH values in test tube L1 and the test tube L4 at the end of the investigation. So L1, okay, so L1 took 25 seconds, L4 did not change color. So for L4 we can easily assume that there was no chemical reaction. So alright, so it will remain blue for L4. So that will be a pH of 8. And for L1 the reaction completes, so it has become yellow. And if it becomes yellow, then it will be pH 6. So the answer for L1 will be pH 6 and answer for L4 will be pH 8. State the variable that was deliberately changed, the independent variable in this investigation. So we deliberately changed the lipase enzyme concentration. State two variables that were kept constant in this investigation. So basically the volume of milk and the total volume of enzyme was kept constant in this investigation. Suggest why all of the test tubes were placed into a water bath in step 5 for 5 minutes before mixing their content. To give enough time for the contents of the test tube to reach the temperature of the water bath. State the potential source of error in step 8. Color change is difficult to judge for the student. Describe how you could safely test lipase for the presence of protein and state the result of positive test. Method. So for protein, we're going to add biuret reagent. And for positive result, we're going to get the color will change from blue to purple. And for safety precaution, we need to wear gloves or goggles. The average temperature of human body is 37 degrees Celsius. Human produce lipase for fat digestion. A student thought that lipase would work best at human body temperature. Planned an investigation to find out if 37 degrees Celsius is the optimum temperature for lipase activity. So, guys, in this kind of question, we must have a following set of ideas that we must implement in order for answering this kind of question. And if we just apply those, definitely we are always going to get the full marks. So for example, we're gonna mention independent variable, method, dependent variable, constant variable. We're gonna mention about repeating the experiment. We're gonna always mention about safety and precautions. So guys, in terms of independent variable, we can write the experiment can be performed at a range of temperature. Example 27 degrees Celsius, 37 degrees Celsius, 47 degrees Celsius, or 57 degrees Celsius. A constant temperature can be maintained throughout the experiment using water bath. The progress of the reaction can be tested using universal indicator. Same volume of milk, same volume of sodium carbonate, same amount of indicator were used in each experiment. So that part will be our dependent variable. So that part, so that part will be our constant variable. The time taken for the indicator to change color was then recorded. The experiment was repeated at least 3 times for each pH, and goggles can be worn. All right, this experiment is relatively of low risk, so this particular points will be enough to get you the required number. Question number 2. Figure 2.1 is a label diagram of parts of a flower. We can see the anther, filament, sepal, style, stigma, petals, ovary, carpel, and stamen. Figure 2.2 is a photograph showing the parts of the flower that have been separated. And then it's labeled with A, B, C. Name of the flower part A, B, C. So basically the flower part A looks like the stigma. And flower part B looks like the filament. And C is sepal. We are also asked to write down the number visible. So we can see there is only one stigma and there are 8 filament. 1, 2, 3, 4, 8 filament and 4 sepal. Figure 2.3 shows a photograph of a germinating pollen grain. We can see the pollen tube. We can see the pollen grain. Magnification 350 times. Make a large drawing of the germinating pollen grain shown in figure 2.3. Label the pollen tube. So when doing drawings like this, we must follow several rules. For example, the drawing diagram should be drawn in single line drawing. There should be no shading. There should be correct details and use more than half of the space that is given. Now the drawing. If drawn correctly, your diagram should look something like this. Measure the length of the line XY on figure 2.3. The length of the line XY in millimeter. So basically we would have to use a ruler to measure the length of the line X to Y. In this case, the answer is 33 millimeter. You just need to use your ruler and find out the length in millimeter. Calculate the actual length of the pollen grain in figure 2.3 using the formula. Magnification is equals to length of XY in figure 2.3 divided by actual length of XY. So in this case, we have this particular information, magnification and length of XY given, and we need the actual length. So we're going to write down actual length of XY equals to, then length of XY, which is 33 millimeter, we're going to divide by the times of magnification, which is 350 times. Our answer will be 0.094 millimeter. Some students collected pollen from the anthers of flowers to investigate the effect of two different solutions S1 and S2 on the germinating pollen. Two microscope slides were prepared. Slide 1 had 210 pollen and 2 drops of solution S1. Slide 2 had 250 pollen and 2 drops of solution. 2 drops of solution S2. Every 10 minutes, the student counted and recorded the number of pollen grains that had germinated. The percentage of pollen grains that had germinated was calculated. Figure 2.4 shows an drawing of the pollen grain as seen with light microscope. Germinating pollen grain, ungerminated pollen grain. Okay, so we already got the key. Now moving on. The results of the investigation are shown in table 2.2. Solution S1. So we can see at 10 minutes we had 5, 18, 26, 40, 51 and 51. So up to 51, all right, percentage of germination is experienced. All right, from 50 to 60 we didn't see any increase in solution S1. In solution S2 at 10 minutes we had 3, then we had 8, 18, 28, 36 and 51. So we can see at 60 minutes both solution S1 and S2 had the same percentage of germinations. However, solution S1 has a generally higher percentage of germination until 50 minutes. We can also see that the percentage of germination in S2 did not plateau until 60 minutes. So stay to conclusion for these results. So the pollen germinated faster in S1 than in S2 because we can see always the values of S1 is greater than the S2 until 50 minutes. By 60 minutes, there were no difference between the percentage germination for both solution. The results in table 2.2 are shown as the percentage rather than as the actual number of germinated pollen grains. Explain why this enables a valid comparison to be made between the results for S1 and S2. So there were different number of pollen at the start of the experiment in each slide. Next question. Describe how the percentage germination in table 2.2 was calculated. So basically, the number of germinated, all right, we divided the number of germinated by the total, then multiplied with 100. The students prepared another 3 slides using solution A, B, C and then left them for 60 minutes. They measured the length of the pollen tube in 20 germinated pollen grains. The results are shown in table 2.3. So solution A, average length of the pollen tube in micrometer 190, B 220, C 265. Figure 2.5 shows the grid the student used to plot the graph of their results. Average length of pollen tube slash micrometer. So A, it has been done for us. So B and C will have to do. Use the information in table 2.3 to complete the graph in figure 2.5 by adding the scale for the y-axis, plotting the bars for solutions B and C. So guys, we added the scale. All right, 10 boxes were equals to 50, and basically, according to the A, we just drew the B and C with the relative height. Guys, thank you for joining this particular class. And if you like the video, please you know give a thumbs up. All right, and comment on the video which paper do you want us to solve, and also subscribe to the channel. Thank you.